I'm Sally Powers. I'm director of the Center for Research on Families, and we're thrilled to have this speaker, who will be introduced in one moment. I wanted to just let you know that this lecture series, which is focused on stress and specifically trying to understand a bit about the physiology underlying stress responses, is sponsored by the center, but it is also part of the Tay Gavin Erickson lecture series within the Center for Research on Families. And the impetus for this lecture series comes from our stress research group, which meets at the center and is a group of about 12 faculty from a number of different departments and disciplines, and we work, we come at studying stress both on our own and collaboratively from a variety of different perspectives. And one of the things we decided as a group is to bring in experts who could really talk with us, have an ongoing conversation, and inform this multiple disciplinary perspective on stress. So we are really thrilled to have Dr. Tom McDade here. But Lynette Leidy-Sievert is a professor from anthropology who's a member of this group and is going to introduce the speaker.
So I am very pleased and thrilled to be introducing Dr. Thomas W. McDade to you this afternoon. So I have known Tom for about 10 years, maybe longer, through the Human Biology Association and the American Association of Physical Anthropology. And Tom is currently on the executive committee of the Human Biology Association. Tom received his bachelor's in biosocial anthropology from Pomona College. And I'll just take a minute to tell the medical anthropology or evolutionary medicine students who are here that he worked with Jim McKenna with the breastfeeding and the co-sleeping and all of that. So there's lots of nodding heads out there. So we enjoy that work. His Ph.D. is in biological anthropology from Emory University. He joined Northwestern University in 2000 as an assistant professor of anthropology, as fellow in the Center for Reproductive Science, and as director of the Laboratory for Human Biology Research. In 2004, he became a faculty fellow in the Institute for Policy Research. In 2006, he became the associate director of Cells to Society, the Center on Social Disparities and Health. He was promoted to full professor this year and is the Martin J. and Patricia Koldyke Outstanding Teaching Professor at Northwestern University. In 2004, he received the Presidential Early Career Award for Scientists and Engineers from the White House Office of Science and Technology. Currently, he is the PI or co-investigator on 7 federally funded research projects, including a study of healthy aging among Filipino women, a study of inflammation in lowland Bolivia, and a study of social influences on early adult stress biomarkers. He is the author or co-author of about 100 publications. So Dr. McDade is a leading researcher in applying biocultural perspectives to better understand health and human development. His work has taken him to Samoa, Ecuador, Bolivia, the Philippines, as well as his adopted home of Chicago. In particular, he studies stress, immune function, inflammation, and has developed a number of the important field and laboratory methods that we use in the study of human biology. So we're just very happy to welcome you here to UMass Amherst. Thank you.
Thank you. Thank you all very much. Thanks for the invitation to be here. It is a pleasure, an honor to be here with you. UMass Amherst is such a storied tradition in human biology and biological anthropology, and I know many of you here, at least by reputation. And so it's a pleasure to see some familiar faces as well as to meet some new people in the Center for Families and the Stress Working Group that put me through some paces this afternoon over an extended lunch, which was really a pleasure. So it's wonderful to be here and share with you some recent work that focuses on inflammation in the context of ecology and development. and worked some stress in there because of the stress working group here. And so what we're going to do for the next 45 minutes or so is I'm going to take you through this story, see if you're convinced. I will definitely leave time at the end for questions. If there are questions along the way, feel free. If I feel like we're getting derailed, then I'll ask that we just move on. But feel free if there's something that I'm not being clear on to pose a question along the way. Could I dim the lights up here? Would that work? Yeah, okay. And can everyone hear me okay? All right, great. I know at least those of you in the stress working group are very interested in how early life environments affect health and disease across a life course, and many of you have probably heard of the Barker hypothesis, or developmental origins of adult disease, which over the past probably 10 to 15 years, I think has really revolutionized our understanding of human biology and health as a life course and even intergenerational phenomenon. But we've known actually since the 1930s, that through cohort and epidemiological data, that environments early in life matter to potential and health and well-being later in life. And most recently, this has really caught public attention as well. So I think it's an exciting time for life course research on human biology and health. And we've always understood health in adulthood to be a product of individual differences in genetic endowments, as well as our lifestyle and the environments that we live in. But now we're coming to appreciate the particular importance of prenatal nutritional environments, postnatal exposures, nutritional and other exposures, and how these have programming effects, lasting effects on various physiological systems during critical periods of development. And we've had that model for the nervous system for a long time, but we're increasingly coming to understand the importance for cardiovascular and metabolic processes and diseases in particular. And a lot of this work in public health really focuses on the sort of long-term damaging effects of suboptimal environments early in life. But in anthropology and in ecology and life course approaches to human biology more general, we often think of this as developmental plasticity, as there is some range of variation in response to environments that may or may not be optimal with respect to long-term things like cardiovascular disease. Most of this work has focused on cardiovascular disease, diabetes, and longevity, but there is a subset of research that's focused on immune development and regulation. That's what I'm going to address today. And that work has really focused mostly on risk for allergy, asthma, autoimmune diseases, diseases where there's something in the immune system that goes awry. And there's a lot of mostly epidemiological evidence, but some experimental, to suggest that aspects of the nutritional and microbial environment early in life matter to your risk for asthma, allergy, and autoimmunity later in life. You may have heard of the hygiene hypothesis. That's what this is. I'm particularly interested in extending this to inflammation and how environments early in life matter to inflammation. And I'm interested in that because I'm interested in the immune system, and inflammation is an important part of innate immunity, as I'll talk about in a minute. But also, inflammation seems to matter to just about everything we care about with respect to diseases of aging. And so I don't know if you missed it, but inflammation is the secret killer. It's going to do us all in, either through heart attacks, cancer, Alzheimer's disease. Basically, you name it. It's hard to find a journal article that has to do with some issue of human health that doesn't have an article or two about inflammation. It just seems to be everywhere. And it's really become somewhat pathologized as this process that contributes to negative health outcomes, particularly associated with aging, which I find very ironic, because if you don't have a functioning inflammatory system, you die. I mean, you just cannot mount a simple response to benign pathogens in our environment. So it's a critical part of host defenses, but that's not what we're talking about now, at least in health research. And this is the observation, the insight that really started a lot of this, moved the field sort of in this direction, and this may be familiar to many of you. Paul Ridker out of Harvard and colleagues are the ones who have been most aggressively promoting this idea that chronic low levels of inflammation contribute to the development of diseases of aging. And this is a very simple but I think elegant demonstration of the power of measuring inflammation. And what it shows you is that you have quintiles of high sensitivity C-reactive protein, a biomarker of inflammation that's commonly measured, which I'll talk a lot about shortly. And if you measure it at low concentrations that are lower than what we previously thought to be significant for health, you find that as you have higher levels of CRP, you increase your risk for heart attack. This is in otherwise healthy people. This is a baseline blood draw. These people were followed prospectively. And independent of where you were in terms of your lipid profile, shown here by ratio of total cholesterol to HDL, there's an additional explanatory power associated with CRP. So people with the highest risk had high CRP and an adverse lipid profile. So from this perspective, there's an argument to be made that we should measure CRP clinically. It provides additional diagnostic information. And then this is what the sort of chronic low-grade inflammation model looks like. And this and a couple other studies have provided justification for measuring C-reactive protein in particular in epidemiological and clinical settings. And what you have here is four individuals who had their blood drawn every 4 days or so over a 7-week period. And the lines just connect the dots of observations within the individuals. So we have 2 individuals here who seem to have pretty low concentrations of CRP. There's one little blip here. And then 2 individuals here that have consistently higher levels of CRP. So there's pretty good between individual variation and not very much within individual variation. And from a clinical chemistry or epidemiological perspective, this is great. You can measure person A's CRP once and know where they're going to be for the subsequent 6 months and compare that to person B and say A is higher than B. Do they have higher risk of cardiovascular disease, something like that. So that's the model of chronic low-grade inflammation. And then in terms of pathophysiology, the idea is that adipose tissue in particular is a very potent and important source of pro-inflammatory cytokines like IL-6. Also, chronic infections, subclinical infections, herpes viruses, periodontal diseases can upregulate inflammatory processes. They promote the production of these inflammatory cytokines, which go to the liver and induce cells in liver to produce and release C-reactive protein, which then goes into periphery and has a number of atherosclerotic effects in tissues in the periphery, particularly in the heart and potentially in the brain, that leads to the progression of atherosclerosis. And this is hypothesized to be an independent pathway to heart attack or stroke from more traditional processes associated with lipid accumulation and blood pressure. There may be some interaction, but the proponents of this hypothesis suggest this is a direct causal effect of inflammation on the progression of disease. Okay, so what do these two have in common? This gentleman, from the look around the middle, probably has pretty high concentrations of CRP due to visceral adipose tissue production of IL-6. This is a horseshoe crab, an organism that's been on the planet since before the days of the dinosaurs, and it also has high concentrations of C-reactive protein. It's a very important part of its innate immune defenses against bacteria and other pathogens. And the species like the horseshoe crab don't have fully elaborated specific immune defenses with lymphocytes like mammals do. But our specific immune defenses were built on a more generalized innate immune system, of which CRP is a critical component. In fact, CRP was discovered in 1930 for its ability to bind to the C polysaccharide region of the Streptococcus pneumonia bacterium. So that's why it's called C-reactive protein. It reacts to that part of the bacterium. So it's an important part of innate immune defenses against infectious disease. And our response, the acute phase response to infection, looks something like this. So this is a different model than the chronic low-grade inflammation model. This is the acute phase model, where basically CRP and other pro-inflammatory mediators are basically undetectable in serum if nothing's going on. And then you get challenged with the flu or a bacterial infection, and production of CRP and other acute phase proteins increases dramatically within hours and increases concentrations in circulation by orders of magnitude. And that C-reactive protein and these other proteins are important for providing a first line of defense, a rapid line of defense against infection. And then as the infection starts to resolve and clear, your concentration of CRP declines, and then you come back to baseline, which is essentially 0. So it's a very different dynamic model of how inflammation happens. And from this perspective, you have a pathogenic challenge that induces the production of pro-inflammatory mediators like IL-6, which incidentally cross the blood-brain barrier and cause the symptoms we associate with infection, like fever and loss of appetite. The liver produces CRP, which then activates complement, promotes the activity of phagocytic cells, which engulf microbes, and opsonizes or tags microbes, identifies them as foreign, so other cells can help clear them from the body. So this is a very important process related to fighting infectious diseases. So we have this situation where we have an inflammatory pathway that is activated by two distinct domains of pro-inflammatory stimuli. There are others, but these are two that I'm most interested in today. And they share common pathways. And we know that CRP matters to innate immune defenses, but we're not particularly interested in that these days, seems like. We're more interested in its potential connections to CVD. And I think in part that's because we only study it in people like this. And because we live in such a privileged epidemiological environment with incredibly low levels of infectious disease, we basically eliminated our ability to study this pathway here, right? We're only focusing on that pathway and its links to cardiovascular disease. So if we're interested in a system, inflammation, that seems to be fundamental to some disease processes we care about, I think we really tie our hands by not studying it in a more diverse range of environments that humans live in and that this system likely evolved in. And so this is why I'm interested in working places like the Philippines, which we're going to go to now, and lowland Ecuador, which we'll go to in a few minutes. So historically, the Philippines has undergone an epidemiologic transition from high levels of infectious diseases and low levels of chronic degenerative diseases to higher levels of things that we all worry about here and lower levels of infectious disease. But there are still pretty high levels of infection in the Philippines, much higher than the United States. And in the study I'm going to tell you about, which began in the mid-'80s, there were still pretty high levels of infectious disease. So it's a different epidemiological environment than the United States, but not radically so, but still significantly different. The study I'm going to talk about today, the data come from the Cebu Longitudinal Health and Nutrition Survey, which began in the 80s by Barry Popkin, economist at UNC Chapel Hill, and Linda Dare joined shortly thereafter, and my colleague at Northwestern, Chris Kazawa, and I are investigators on the study now with Linda. And the study started in 83, 84, just about the time I was starting high school, and with the recruitment of 3,300 women from neighborhoods that were randomly selected into the study. And an attempt was made to recruit every single woman who was pregnant at that time into the study, so it would be representative of those communities. And they were assessed for measures, anthropometric measures, aspects of lifestyle, the household environment, community environment that they lived in. They were followed up at the delivery of their babies. The babies were weighed. And then every 2 months subsequent to that, the moms and the babies were visited. The babies were weighed. Information on breastfeeding, growth, infectious disease was collected. And then that was supposed to be the end of the study. It was designed as a study of maternal and child health and what factors could promote breastfeeding in impoverished environments at a time when formula feeding was being aggressively promoted internationally. And it provided a lot of very important evidence to show the benefits of breastfeeding for child survival and positive growth outcomes that was really used to fight a lot of the formula campaigns at the time. And that was it. But then opportunistically, there were a couple follow-ups. And this study is done in a really close partnership with colleagues at the Office of Population Studies at the University of San Carlos in Cebu. And we're both on equal footing with the study. And they were interested in following these people up, seeing where they ended up. And about this time was when there started to emerge a lot of interest in the early origins hypothesis, the Barker hypothesis. And so we realized, this is my postdoc right here at UNC Chapel Hill with Linda, that this was probably the best data set in the world, if not the best one of 2 or 3, for looking at how environments early in life matter to physiological function and health later in life. So in 2005, we designed a study to find as many people as we could, and we collected all sorts of life history information, educational, sociodemographic, psychological information, and also collected blood and saliva samples, which we measured for many things, only one of which I will really talk about today. All right, so what does CRP look like in the Philippines compared to the United States? The red bars are Filipinos. This is 2005 data. The kids in the study were 21 or 22 years of age. And for comparative purposes, I drew age-matched data from NHANES, which is nationally representative United States data set. And what we see is that overall, the median CRP is about 0.2 or 0.3 milligrams per liter in the Philippines compared to 0.9 in the United States. and there is a slide of sex differences here. I forgot, this is the only one though. That there are no sex differences in CRP in the Philippines, but consistently reported in the US, women tend to have higher CRP than men. So I've said that the Philippines environment has higher level of endemic infectious diseases. You might have expected higher CRP there than in the United States. That's not the case. But one obvious possibility, given what I said about the contribution of body fat to inflammation, is that Americans are bigger than Filipinos, which is true. The average waist circumference in the Philippines is a lot smaller than it is in the United States for young adults. So it could be that Americans have more visceral fat and therefore produce more IL-6 and therefore produce more CRP. Well, if you break it down by waist circumference groups, within every group, Americans still produce more CRP than the Filipinos do. So it's not just differences in body composition or overall body fat. There's something else going on that has to account for these population differences in C-reactive protein. So what might that be? One question that I used to get a lot, and then therefore I put this slide in, is that, well, there must be genetic differences between the populations. And there are several genes that have been identified that are related to the production of IL-6 and CRP and other pro-inflammatory mediators. And just, it's my good fortune to have a colleague at UNC Chapel Hill, Karen Mulkey, who's interested in genetics of obesity and diabetes, who was involved with this study and does a bunch of parallel work with Linda Adair. And she analyzed the DNA from this study and looked at all the SNPs that are related to inflammation. And they only explain a very small fraction of the variation in CRP in this population, about 5%. And the pattern of associations is very consistent with what we see in American and European and other Asian populations. So genes can't explain this phenomenon at all. So I love that slide just to be able to say that that's not it. So then, of central interest to me, do early environments shape the regulation of inflammation in adulthood? Could be that, is there something about environments early in life there, both compared to the US and also variation within those environments, that could shape set points, regulatory set points that carry forward into adulthood with respect to inflammation? So we know that among adults, the amount of body fat you have and your level of pathogenic exposure both matter to your CRP. So might there be analogous exposures early in life that matter to this developing system that then have independent or perhaps interactive contributions to CRP in young adulthood? A priori, there are very good reasons to expect that the first couple years of life will be critical for the development of the immune system. What this figure shows you is by age, so starting at birth going up to adulthood, age 20 here. The top figure is enumerative measures of immune function, so numbers of T and B lymphocytes, the size of the thymus, concentration of key immunoglobulins. And these are functional measures. How likely are your T cells to proliferate? How active are your NK cells? And in almost all cases, except for immunoglobulins and monocyte chemotaxis, things are really active in infancy. And there's good reason for that. Your immune system comes into this world sort of hungry for engagement with its environment because it needs to learn quickly about the microbial threats that could be mortal. So the system is sort of on overdrive, primed to engage with the environment and learn from the environment. So there's good reason to think that these first couple years of life may be critical to the immune system in general and also to inflammation as part of the immune system. So with our data set, we have information about the gestational environment, measures of birth weight and ponderal index, which are used commonly to indicate the quality of the nutritional environment prenatally. Imperfect measures, the best we have, but used very commonly epidemiologically. Preterm delivery or not parity, and multiple measures of socioeconomic status at the individual household and community level. And then in infancy, we have measures of growth, patterns of breastfeeding, cleanliness and pathogenicity of the household environment, and frequency of infectious disease symptoms like diarrhea and respiratory infection. And then in adulthood, we have parallel measures of anthropometry related to body composition, infectious disease, health behaviors, things that have been shown to matter to CRP in adults that we can control for to hopefully identify the more direct effects of the early environment factors. All right, so this is what we find with respect to birth weight. Individuals born with higher birth weights have a lower probability of having elevated CRP in young adulthood. And I'm defining elevated CRP here as being in the top third of that local distribution. And I can tell you why I use that point later if there are questions. So birth weight matters. And this was the first study to show that individuals born underweight or lower birth weight have higher likelihood of inflammation later in life. And this is potentially important because there are a lot of studies showing that lower birth weight individuals have higher cardiovascular disease risk. And this could be a mechanism contributing to that. So that's great. But it can't explain the low levels of CRP in the Philippines compared to the United States, because Filipinos on average are smaller at birth than Americans are. Okay, so this is interesting, potentially important, but doesn't address the broader issue of regulation and population differences in inflammation. This, to me, is much more interesting and I think potentially informative. We found that individuals who had higher levels of infectious diarrhea in infancy had lower CRP as young adults. All right? So this is not a complete census, not a complete accounting of infectious diarrhea in infancy. Remember, we went every 2 months to the moms and interviewed them and asked them if the week before their kid had had any symptoms of infectious disease, in this case diarrhea. And the higher number of intervals where a mom said, yes, my child is or was sick over the past week, is associated with lower concentrations of CRP in young adulthood, independent of everything else that we can throw at this model to try to explain this away. Okay, so that's one. Frequency of fecal exposure. So this is a tropical environment. Most of the homes are, at least at the time in the 80s, open air. And a lot of them were suburban or somewhat rural. And even many of the urban homes, domestic animals were allowed to roam freely in and through the homes. So the interviewers at each of these bi-monthly intervals would observe whether animal feces were present, could be visible, or evident on the floor of the home, and whether the baby was at a stage that he or she was crawling on the floor. So it's a proxy measure of likelihood of contact with animal feces. And the higher frequency that that was true at each of these intervals, the lower CRP as a young adult. And then lastly, we have season of birth. So the Philippines is a highly seasonal tropical environment. There's seasonality in rainfall and in pathogen transmission. And a lot of the early work with this study documented seasonality in rates of infectious diarrhea. So we have a pretty well-validated handle on differences in exposure during the dry and non-dry seasons. Kids who were born in the dry season actually spent more of their first year of life in the non-dry season, which is when more infectious microbes were in their environment. So if you're born in the dry season, you had higher levels of infectious diseases in your first year compared to someone born in the non-dry season who spent more of their first year in the dry season. All right? So dry season birth, more infectious exposures, lower CRP as a young adult. So we have, I wouldn't put too much stock in any one of those, but all three converge on this conclusion that low levels of microbial exposure, independently from low levels of nutrition prenatally, are associated with higher CRP in young adulthood, independent of pathogenic exposures in adulthood and level of body fat in adulthood. And so we don't know exactly what the mechanisms are. It could have to do with cytokine signaling. I can talk about that more in a minute. But at least I have convinced myself that there is something real going on here with microbial exposures in infancy that matters to the regulation of inflammation. All right. Conceptually, this is how I think about this. Okay? So I'm going to start to build a model and convince you it's true with some data from Ecuador, and then share some implications of that if it's true. So we can see if you are convinced or not. In a higher pathogen environment, in infancy, you have lots of microbial exposures. These red arrows are hits, stressors on the system or inputs into the system. And this acute phase model is such that you'll have an acute inflammatory response, and then you'll shut it down. And that happens repeatedly. And so you have a lot of opportunities in that environment for the system to learn how to fire itself up and turn itself off efficiently. And then as an adult, these kinds of exposures are less frequent. You're not crawling on the floor. You're washing your hands. And your immune system is better able to deal with microbial threats. So these kinds of inputs are less frequent, but when they happen, you can deal with them effectively, efficiently, ramp things up and turn things off quickly. If you are in a low pathogen environment, a very hygienic, highly sanitized environment, these opportunities come infrequently, and they may not be as intense. And so you have fewer opportunities for engagement with the environment in such a way that your system learns how to regulate itself. And that may carry through into adulthood. And then when you have pro-inflammatory stressors in adulthood, the system is slow to turn on, or perhaps, I think more importantly, inefficient at turning itself off, and you get a pattern of overblown or chronically activated inflammatory processes. All right. So we can start to think of it like this from a life course perspective and how early environments and inflammation may matter to disease in adulthood as such that your early life microbial and nutritional environment helps determine what kind of inflammatory phenotype you develop as a child and through adolescence. One like this that's sort of tightly and efficiently regulated, one that's more diffusely and poorly regulated. And then as an adult, this shapes the level and dynamics of inflammation, and that has implications for disease. So if you have this more tightly regulated, efficient system, I would hypothesize that you in general would have low CRP, but you could have acute elevations in CRP, very high levels of inflammatory activity, but only in response to a challenge, and then you shut things off when you don't need the system. And under that scenario, I would not hypothesize that there would be a contribution to cardiovascular disease in the way that people are talking about it right now. Under this system, under this scenario where the system is more diffusely regulated, you have high chronic CRP, but also you can mount acute elevations in response to the flu or something like that. And under that scenario, inflammation very well may contribute to the development of cardiovascular disease. Yeah?
Curious if you're suggesting these microbial exposures have to be pathogenic or even mildly pathogenic, because I think some of the same rules have seen probiotics in the bacteria that are not pathogenic.
Yeah.
Or even opportunities.
Yeah, yeah.
Is it nutrition, or is it confounded here that the one that, animal crap on the floor is eating different food, or is it actually exposure?
Well, you kind of went somewhere else at the end there than I thought you were going. But the first point is, does it have to be pathogenic? And I think the answer is no. What I think is lacking, or what I think is driving this, is benign microbial exposures to common microorganisms that have been part of the human environment forever. Things that never result in a clinical infection, but that are important in their diversity and intensity for training up the immune system. So I've actually tried to transition my language away from using that word pathogen, although it was in that last slide, to microbe, because I think that's what's important. So there could be probiotic things that do this, that help train up the immune system. With respect to nutrition versus microbial exposure, I mean, this is kind of a sledgehammer approach to this question using available data. So we don't have very fine measures of microbial exposure. I can't even tell you if it's bacterial or viral that really matters. I have some ideas, but we just don't have very high resolution about diet or the type of microbial exposures that these kids had. So that's a limitation. All right. So now that's the model. That's sort of the working model. And now I have some evidence I think supports this model. So this is, this study in particular, Macy et al., and there's one other, that are used as justification for this chronic low-grade inflammation model, both in terms of measurements and in terms of modeling inflammation. So this is the same study that I showed you before with the 4 individuals over a 6-month period, over a 7-week period or whatever it was. This is 26 individuals over a 6-month period, and the plot is a little bit different. This is subject number. So these are individuals on the line here, and each dot is a CRP value. And so each individual has 4, 5, or 6 values, and the line connects those. And so what you can see is that people tend to cluster, right? And this cut point here, 3 milligrams per liter, is commonly used as the cutoff value for high risk of cardiovascular disease, right? And that's been in play for about 6 or 7 years based on a Heart Association CDC study. And so there are a subset of individuals that produce all their CRP values above that value. And then there's another subset who are close, and they're clearly higher than these other individuals. So again, a high degree of between individual variation, and some within individual, but there's more between individual. So as an epidemiologist or even as a clinician, I could come in and take one of these samples and say, okay, you are definitely at higher risk than this person. And this is why this is being promoted clinically and epidemiologically as a useful measure for chronic inflammation, even with one blood sample. The problem with this, I mean, it's not a problem. This is a very good study, and I believe that this is true. But everything we know about variation in inflammation and CRP biovariability in particular has been done in the United States. in this privileged epidemiological environment. So given what I said about the development of the immune system and how it may be responsive to the microbial ecology early in life, you might hypothesize that this could look different in a different epidemiological universe. So I pitched that to NSF. I was very happy to get funded to do this study in lowland Ecuador, Amazonian Ecuador, which we completed last summer as part of the Shuar Life History Project in collaboration with folks at the University of Oregon. And I actually did not go to Ecuador. I had a graduate student who was very eager to go, so she went and collected these data for me, and I was analyzing them yesterday and putting these slides together today. I was sending to the stress working group. I was very excited and motivated to get this done in anticipation of this talk. And so we recruited 52 adults into the study, and we visited them every week for 4 weeks and collected a blood sample. And we use finger stick blood sampling, where we put blood on filter paper that dries and preserves the blood. So we can get blood even in the Amazon and ship it back to my lab and analyze it, and things are stable, and we validated all of that. So it allows us to sample repeatedly without too much inconvenience to participants. And this is the pattern that we get. So it's just like the plot before. These are 52 participants, 4 observations for each, and they're sorted based on the average of these 4. And do you see a difference? It's pretty dramatically different, right? There are none of those. There's not a single one. About a third of the people produced an observation above CRP, but above 3 milligrams per liter, but every other observation for those individuals was less than 3. So you can, in this setting, the individuals could produce a high value, but they also could produce a low value. So look at the floor here. Almost everyone has a CRP value that's less than 1 at one of these intervals. So another way to look at it is this. This is a plot across time, 1, 2, 3, 4 weeks, just for the 17 individuals who at one point produced a high CRP value. And if you were doing a cross-sectional study at, say, sampling interval 2, you'd say, okay, I have 1, 2, 3, 4, 5, 6 people have high CRP. And the prevalence of chronic inflammation is 30% or 20%. But if you went back the next week, every single one of these individuals had a value that was less than 3. So basically, there is no chronic inflammation in this setting. Not a single person meets the criteria promoted by this AHA CDC statement for chronic low-grade inflammation based on this study design. So I think this has implications not only for measurement and study design, especially in places with high infectious disease burdens where, incidentally, 3/4 of all cardiovascular disease cases are globally, but it also has implications for how we think about inflammation, which has implications for how we model it. One other piece of evidence in support of this model, perhaps, is that we've looked at, just very recently, concentrations of pro- and anti-inflammatory cytokines in the Philippines study that I was telling you about. And I just published a paper where we reviewed what cytokine concentrations in the Philippines look like, these are the red values, compared to all the previous published literature from healthy individuals, which was hard to find. There's a lot of research on cytokines, but it tends to focus on clinical cases and pathological cases. And we have among the lowest levels of IL-6, that pro-inflammatory cytokine, and among the highest levels of IL-10, which is a very potent anti-inflammatory signal. It actually prevents IL-6 from activating pro-inflammatory pathways. So the balance of pro to anti-inflammatory cytokine signaling seems to be different in our sample from the Philippines than in prior studies. So to me, that is further evidence that suggests there is something different about how early environments can shape the regulation of inflammation in adulthood. Okay, some implications and some hypotheses here. So if we have this model that early environments matter to your developing immunophenotype, how you regulate inflammation into adulthood, a lot of the things we're interested in relation to inflammation are inflammatory stressors, events that matter to inflammation. And instead of just thinking about how these things have a direct effect on inflammation, we might want to think about how early environments set up the dynamics of the system in such a way that they moderate your response to these inflammatory stressors. And so infection is a pro-inflammatory stressor. Pregnancy is actually a mild pro-inflammatory state. I'm going to show you some data on that. Vaccination is a nice model for activating inflammation, and then you can look at individual differences in in response. Body fat, smoking, psychosocial stressors, these are all things that have been positively associated with inflammation. But how you regulate inflammation may modulate your responsiveness to these pro-inflammatory stimuli. Does this provide some insight into this scenario? Could it be that adipose tissue is indeed a pro-inflammatory input into the system, but if you are in an environment where you have a better established anti-inflammatory regulatory network, can you keep that inflammation in check despite the same level of body fat as someone in another environment? So to me, this provides some at least, I don't know, conceptual explanation of this difference in the relationship between body fat and CRP between the Philippines and the United States. I know many of you are interested in depression, and Andy Miller is coming next week. I wish I could be here for his talk. When I was at Emory, he was just getting started there, and I don't know if he'll remember me, but at one point in my middle of my graduate career, he gave me some very good and important advice. So you can tell him I said that and see if he knows what it is. So there's a lot of interest in the bidirectional relationships between depression and inflammation. Pretty strong evidence to suggest that it matters in both directions. Inflammation, cytokines can cause depression, as well as depression causing increases in inflammation. So these are just a couple of studies showing that. We have information on depression in our Philippines study. Let's leave aside for a moment whether depression is the same thing and whether we measured it correctly in a Filipino context. Let's just forget about that. But let's assume that I have some valid measure of depression in this setting. I can't find any evidence for an association between depression and these three inflammation-related markers there. So there's nothing there. So why is that? Is it that the dynamics of the system are set up differently in such a way that they're not sensitive to this inflammatory input? I don't know. That's something I'd like to investigate further. There's also been a lot of interest in stress, aside from depression, perceived stressors, objective stressors, and their contribution to inflammation. And I published a paper a few years ago in Chicago showing that perceived stress among older adults is associated with higher levels of inflammation. Maybe a mechanism through which stress matters cardiovascular disease risk. So I wanted to look at that in the Philippines cohort, and I do find a mild positive association between perceived stress. This is just using the Cohen perceived stress scale. And so individuals who report higher levels of burden and less control and more stress in their lives have higher CRP, but this is not a statistically significant association. But what if I stratify by an early life microbial exposure variable? And so I did this with dry season and wet season birth. So remember, being not born in the dry season means lower levels of infectious disease your first year of life. And we get an association that's more consistent with what's been reported in the United States. Higher levels of perceived stress, higher levels of inflammation. Born in the dry season, which means higher microbial exposures in infancy in that first year, there's no association. If anything, it's negative. And so if you look just at this difference here, the effect, if it is an effect of high stress, is much different based on your early life environments. So this, I just pulled this together a few days ago. So this is something that I need to follow up. All right, so then this will be my last example. We have some really cool preliminary data. One of the things that's great about this study is that because it's been such a great partnership with our colleagues in the Philippines, and because actually the participants have become quite invested in the study, they see its value. They've established great relationships with the field workers and our colleagues at the university there. And they are now in their mid-20s and having babies of their own. And so this study is now becoming a 3-generation study because we have the original moms, the kids that are now becoming parents, and their babies are now in the study. And no one has said no to that among the people who are still around. And in 2005, when we collected blood, there were several women who were pregnant, almost 100, about 80-some who are pregnant. And we've just sort of been excluding those values from our analyses because things are different in pregnant women. But that's an opportunity to actually look at some variation in inflammation, especially since pregnancy is an inflammatory event at some level. So just forget about this for a second. This is just the distribution of patterns of inflammation and regulation among this subset of women who were pregnant in 2005. And about a quarter of the women had low CRP. And this is all just median splits because the cell sizes were quite small. about a third of the women had high CRP and low IL-10. And I'm hypothesizing that this is a sort of out-of-control inflammation situation. So you have the biomarker, the endpoint, that is up, and you don't have very high levels of the anti-inflammatory signal. So I think of this as cruising down the highway, but the brakes aren't really working. Okay? And then about another third of the women, a little more, have high CRP, but also have high IL-10. So to me, this is cruising down the highway really fast, but you can slow down really quickly if you need to. It's a well-controlled inflammatory state. This was the distribution of these women who had low levels of microbial exposure in infancy. And this, because of the sample size, again, I just put together a set of variables to represent overall microbial exposure and just split it in half. Women who had high microbial exposure in infancy, almost twice as likely to be in this well-controlled inflammatory state. And the reason why I think this might be particularly important, and this line of reasoning actually came from this article, which I happened to stumble across a couple of years ago, which showed that the impact of inflammation on death following heart attack was totally dependent on the level of interleukin-10, IL-10. So this is a study that recruited people after they had a heart attack, followed them up for 6 months or a year, and then the endpoint was death or another heart attack. And so at baseline, there were individuals with high and low CRP, and the individuals on average with high CRP did worse, but only if they had low IL-10. The people who did best over that period of follow-up were people who had inflammation, high CRP, but high IL-10, so a controlled inflammatory state. So again, this suggests to me, at least, inflammation is good for us. I mean, it serves some purpose, but we have to control it because it's potentially damaging as well. So might this be a similar scenario with pregnancy? And do these women who have the well-controlled inflammatory state have better birth outcomes? And with our sample of 80-some, the answer is yes. So we have birth weights on this next generation of babies. The best birth weights are to women who have controlled inflammation during pregnancy. The worst birth weights are to the women who have inflammation that's out of control, or at least that's my interpretation of it, as being out of control. So we use this as justification for a grant application that just went in last spring and just did not get funded. So, but we're going to try again. Okay. So I'm just going to point to some future directions here, and then we should have plenty of time for conversation. I think we can start to think about health in general as a life course and even intergenerational phenomena, and inflammation may be a particularly important part of this process. So here's the life course. Here's generation 1. And we know that the nutritional, infectious, and psychosocial environment, which I haven't talked so much about, but there's evidence to suggest the psychosocial environment, particularly in childhood, matters to inflammation later in life. These things are all related to aspects of the social, economic, resource environment. They shape your inflammatory phenotype, which then helps define your risk for inflammation-related diseases of aging, but also has impact on the next generation through its impact on inflammation during pregnancy. And so we have this cycle across generations. And so we're trying to test this model in 2 different ways. One is a new study that is just gotten off the ground. I'm going to the Philippines in December to help launch. And this is with the moms. So these women are in their 40s, 50s, 60s now, and they're aging into higher risk for cardiovascular metabolic diseases. And so we want to know if their inflammatory phenotype affects their risk for inflammation-related diseases, like it has been shown to do in the United States. In addition to measuring CRP and IL-6 just in sort of baseline samples, we're going to give a subset of women a flu vaccine, which is something they want, and we're going to monitor their inflammatory response to the vaccine. So we can actually differentiate between this pattern of regulation and this pattern of regulation and see prospectively if that predicts cardiovascular disease risk. So that's an important test of one of the key assumptions of the model. The other thing we're doing is trying to look at the impact on the next generation in those kids. So this is analogous to the preliminary data I just showed you. All the blue boxes are data we have in this study. So we have information on generation 2 who are having babies right now, right here. I'm sorry, generation 1 in this scenario. We have information on their prenatal environment, their postnatal infancy environment, We have their CRP and IL-6 and IL-10 in young adulthood. And we have some blood samples from them during pregnancy. So we can look at inflammation in pregnancy and how that affects the environment of the next generation. We're also proposing to look at some upstream measures of gene expression related to inflammation, and also some epigenetic modifications that may represent a lasting imprint of those early environments on these regulatory processes in young adulthood. And to me, what's most compelling and interesting about this is that most research on inflammation and its contribution to disease focuses on adults and what kind of level of inflammation they have and how that matters to their risk for diseases of aging. But we really need to understand that inflammatory phenotype in relation to earlier environments in that person's life, but also perhaps environments of their mother, of the preceding generation, that shaped their earliest environmental exposures in utero and in infancy. So this is a real life course, transgenerational approach to the study of health, and inflammation may prove to be an important mechanism as part of that process. So these are some of my key collaborators. Some of you may know them, and some of the support that we've gotten for this project. So I hope we have some good conversation. I'm happy to answer questions.